This is the fourth and final video for this week's topic of neuroanatomy. Now we'll be looking at the structure of muscle very briefly, the role of the motor unit, as well as neurological disorders. To start off, I just wanted to go over the macrostructure of muscle. Going from right to left, we have the muscle belly, which is attached to tendon. The muscle belly itself is surrounded by a layer of connective tissue called the epimysium. This is also referred to as the fascia. So for those of you who may have gotten some type of deep tissue massage, fascial release is something that can be performed, and that's essentially what we're referring to here. If we go a step deeper, then we have something called the fasciculus, which is simply a bundle of muscle fibers. Now, if we look at each individual fasciculus, they are surrounded by another layer of connective tissue called the perimysium. So if we pull out one of those muscle fibers from the fasciculus that's protruding from the muscle belly, each of these fibers are surrounded by another layer of connective tissue called the endomysium. And just to be clear, a muscle fiber can also be referred to as a muscle cell. Those terms are interchangeable. The muscle fiber, or muscle cell itself, has its own cell membrane. It's called the sarcolemma. It's just a fancy way of saying cell membrane for the muscle cell. If we look into the muscle cell itself, it does have multiple nuclei, which is a very unique characteristic of muscle cells, that they are multinucleated. Generally, we just think of cells as having one nucleus, but that's not the case for a muscle cell. And the cell cytoplasm is referred to as sarcoplasm. So again, just a specific term for cell cytoplasm for a muscle cell. And then one of the last components that's pulled out of this muscle fiber is the myofibril. The myofibril is what is composed of our myofilaments, or the actin and myosin filaments that are essentially creating the shortening or contractile properties of muscle, and perhaps eccentric contraction or some type of lengthening. If we look a little bit closer at the microstructure of our myofibril, which is what we're looking at here, we can then look at the orientation of the myofibrils, the orientation of the myosin, which is the red with the tails, and the actin, which is the helical structure composed of sort of spherical proteins. And it's the pulled out section, the sarcomere, that we see at the bottom left of the screen, that is considered the functional unit of the muscular system. And by function, I'm referring to contractile. So it is the smallest contractile unit of the muscular system, meaning this is the smallest unit where we see actual contraction happening. And the myofibril that we saw pulled out of the muscle fiber, well, that's simply sarcomeres in series. It's just a bunch of different sarcomeres that are connected to each other or adjacent to each other. So I mentioned that we can make distinctions between higher order centers as well as lower order centers, and this idea that there is hierarchical organization of the nervous system is definitely occurring, and that is depicted in this figure. Typically, neural activity is following this level of organization from higher to lower centers. So we see the higher centers at the top, and then some of the middle levels, which would be the sensory motor cortex. That's the cerebral cortex. We know the basal nuclei. That actually is another term to refer to basal ganglia. So that is subcortical. The thalamus, that's in the diencephalon, that's smack in the middle. And then we have the brainstem and cerebellum located below the cerebrum. And then now we start getting to the lower levels, the brainstem and spinal cord, is considered local level in this particular figure. The brainstem is still technically part of the higher order centers, and the spinal cord is now where we're getting to the lower order centers. And then we also have receptors as well as muscle fibers. So there's a lot of information flow back and forth between various structures. You can see essentially double-sided arrows between the brainstem and the cerebellum, and we also see double-sided arrows between the brainstem and the sensory motor cortex. And this is really showing us that flow from the receptors moving upwards towards the higher order centers. That is the flow of afferent information or sensory information. Information flowing from the higher order centers down to the level of the muscle fiber, that would be the flow of our efferent or motor information. So how does that motor information actually travel? It travels via, something called the motor unit. This is a structure that is incredibly important in terms of its role for performing voluntary movement. And the motor unit is composed of an alpha motor neuron, because there are different types of motor neurons. So it is specifically an alpha motor neuron and all of the muscle fibers that alpha motor neuron innervates. That can vary quite a bit. And that has to do with the level of muscular control. The number of muscle fibers served by a motor unit determines that level of control. So we can have anywhere from a few, just a handful of muscle fibers innervated by the same motor neuron, which occurs with muscles in the eye, to something like the gastrocnemius, where we have one motor neuron innervating well over 1,000 individual muscle fibers. So in that case, we would have gross control. You don't really have very much control when there are many muscle fibers innervated by the same motor unit, but you do have very fine control with a muscle in the eye where only a few muscle fibers are innervated by the same motor unit. So the motor unit is a general structure, but we do have different types of motor units. In other words, different compositions of motor units. We have type 2B, type 2A, and type 1 motor units. And the characteristics of each of these motor units really is a reflection or determines their functional capacity. And it's the morphological as well as the physiological characteristics that determine their function. The type 2B and type 2A are commonly referred to as the fast twitch muscle fibers. Type 2B can be referred to as fast fatigable because it has very low fatigue resistance, whereas type 2A is slightly more fatigue resistant. And the type 1 is commonly referred to as the slow twitch muscle fiber because the twitch response is fairly slow and low, but it can be sustained for a long period of time. So here's an example that shows us some of the differences in the three motor units in terms of their functional capacity. Twitch is a reference to the amount of force that a motor unit can generate after one brief action potential or one brief impulse or stimulation. The fast fatigable, again, is type 2B. The fatigue resistance is the fast fatigue resistant is the type 2A. And then the slow twitch is the type 1. So you can see grams of force. The units, I'm not really too concerned with the units here, but what I want you to see is just relatively speaking how different one twitch is for a type 2B compared to even the type 2A and the type 1. The middle figure shows us unfused tetanic force. Tetanic force is referring to the amount of force that can be generated when the rate of firing for each motor unit is very high. In other words, if you're trying to fire a motor unit, you could have it fire once, then there would be a pause, then it fires again, then there would be another pause. But you can also in another scenario, build upon the previous firing. So if we have it fire and then immediately fire again and immediately fire again, then the force just continues to build up to a certain point. So we see near limits in this middle image of the amount of force that each of these motor units can generate, with the fast fatigable obviously much, much greater than the fast fatigue resistant and the slow twitch. But you can't have your cake and eat it too. You can't generate large amounts of force and also be able to sustain that for long periods of time. And this is a reflection of the inverse relationship between force production and fatigability with regard to the motor unit. Our fast fatigable or type 2B motor units cannot maintain high levels of force for very long periods of time. The fatigue resistance, fast fatigue resistant or type 2A, they have greater fatigue resistance than the type 2B, but our type 1 can resist fatigue the best compared to any of our motor units. However, the trade-off is that the amount of force that it is capable of generating is not very high. So if we're talking about recruiting human muscle, motor units follow something called the size principle, where motor units are recruited in order of their threshold potential. Threshold potential is referring to the level of electrical stimulation that's required in order for the motor unit to actually be activated and send the neural impulse to another neuron or perhaps to the innervated muscle itself. So what the size principle is telling us is that essentially the smaller motor units, which are primarily type 1, are going to be recruited before the type 2 motor units, which are typically larger than type 1. Because the smaller the motor unit, the lower the recruitment electrical threshold is, whereas the larger the motor unit, the higher or the greater the threshold will be. But as a result, our smaller motor units, which are recruited first, cannot generate as much force. So if you want to generate large amounts of force, typically you do have to recruit the smaller motor units first before the larger motor units, although there are some exceptions to that rule. Motor units also follow another rule or principle called the all or none principle, which means that all muscle fibers in a motor unit contract and develop force at the same time. In other words, once a motor unit is activated, its recruitment threshold potential has been met or exceeded, then the impulse will stimulate contraction of all of the muscle fibers that motor unit innervates. You cannot activate only part of the innervated muscle fibers and not the rest of them, according to the all or none principle. So this figure shows us a sample of 4 different types of stimulation. We have curve A, which is a single twitch. We have curve B, a summation of twitches, and C, unfused tetanus, which is similar to the figures that we saw for the 3 motor unit types before. And then we have D, something called fused tetanus. Fused tetanus is where the frequency of activation of motor units is occurring so high or so quickly that everything smooths out. They build upon each other in an optimal way, and this is how we ultimately get smooth, coordinated movement to happen. And essentially what this figure is showing us are changes in firing rate frequency. In other words, as we go from left to right in this image, the frequency of activation of a particular motor unit is increasing. So those neural impulses are happening faster between each other. In other words, there's less time between activation as we go from left to right. But one thing that you don't see in this figure is the strength of the action potential itself, because that has no bearing on how strong a contraction will be for a given motor unit. So the strength of the action potential does not have any effect on the actual strength of the contraction for a particular motor unit. The last part of this lecture has to do with disorders of the nervous system. There are multiple kinds, but I am only reviewing some of the more predominant types. The first is a disorder that's commonly referred to as Lou Gehrig's disease. It's amyotrophic lateral sclerosis. The symptoms of this particular disorder, I should say, is progressively worsening paralysis until most skeletal muscles cease to function. So it can be very, very destructive. And what is the neuroanatomical basis for this disorder? Well, the motor neurons of the brainstem and in the spinal cords just spontaneously start to die. And the result of this is muscle wasting, particularly the muscles that are innervated by those motor neurons that are dying. So as a result of not being innervated anymore, the muscle cannot be activated and the muscle begins to atrophy. So that is the reason why the progressive paralysis occurs. Another disorder is Parkinson's disease. This receives a lot of attention in the media. Some of the symptoms that occur with Parkinson's are things like akinesia, which is a failure to initiate voluntary movement, bradykinesia, which is a slowness of movement. Tremors are very commonly noticed, rigidity of muscle and movement, and also postural instability. The neuroanatomical basis of this disorder is a progressive degeneration of dopamine-containing cells in the substantia nigra. This is an area that is subcortical, it's below the cortex, and it is a group of cells that project into the basal ganglia. So it is considered to be a part of the basal ganglia, the substantia nigra. And what happens is that with this progressive degeneration of these cells, symptoms begin to occur. And unfortunately, there's a continuous loss of cells in this area over time. And with very extensive degeneration or cell death, that's when symptoms start to pop up and they become progressively worse. The last neurological disorder that I'd like to briefly cover is Huntington's disease. Some of the symptoms are somewhat similar to what we see with Parkinson's disease, but we also have some differences. Chorea is the involuntary jerking or writhing of the body or limbs. Dystonia is sustained muscular contraction. This, of course, is involuntary. Rigidity is also something that occurs. Postural instability, these are two things that we saw with Parkinson's disease as well. Individuals with Huntington's disease also show some other problems, difficulty swallowing and speaking. And then there are cognitive and psychiatric deterioration as well. The neuroanatomical basis of this disorder is similar to Parkinson's disease in that it attacks the basal ganglia, but it's a different part of the basal ganglia. The progressive destruction occurs at the caudate nucleus and the putamen, as opposed to the substantia nigra. We did not cover the substantia nigra when we looked at the structures within the brain, specifically because their role in terms of voluntary movement is not really that significant. So we did not cover that, but obviously knowing where they are in perspective to the other structures is important because there is some role in terms of controlling body function. So this is the end of the lecture for the week. We do have a term paper at the end of the quarter, and these last topics, the Lou Gehrig's disease, Parkinson's, as well as Huntington's disease, are potential topics for you if you are interested in further exploring any of them for that paper. Of course, you're not limited to clinical applications for the topics. You're really very open to choosing whatever type of related concept from the class.